Welcome to the fascinating world of genome-wide analysis. Today, we're diving into the intricate concepts of chromosomes, scaffolds, and contigs. Let's start with chromosomes. These are the thread-like structures found in the nucleus of our cells, composed of DNA and proteins. Humans typically have 23 pairs of chromosomes, which carry our genetic information. Each chromosome is a tightly packed bundle of genes, the fundamental units of heredity, dictating everything from eye colour to susceptibility to diseases. Now, when scientists want to study these chromosomes in detail, they often break down the DNA into smaller, more manageable pieces. This is where the terms scaffolds and contigs come into play. A contig is a sequence of DNA that has been assembled from overlapping fragments representing a continuous stretch of the genome. Think of it as a puzzle piece that fits perfectly with others to form a larger picture. Contigs are essential for creating a draft of the genome because they help researchers identify the order of genes in other important regions. However, contigs alone don't give us the full picture. This is where scaffolds come in. Scaffolds are larger structures that consist of multiple contigs linked together, often with gaps in between. They provide a framework that helps scientists understand the three-dimensional organization of the genome. By connecting contigs into scaffolds, researchers can gain insights into how genes interact and function within the larger genomic landscape. In summary, chromosomes house our genetic material, while contigs and scaffolds are crucial tools that allow scientists to piece together the complex puzzle of the genome. Understanding these concepts is vital for advancements in genetics, medicine, and biotechnology, paving the way for innovations that can transform our understanding of life itself.